Cytokines are soluble messenger molecules usually secreted by cells of the immune system. Some cytokines, such as interferons and tumor necrosis factors, are secreted by non-immune cells, such as epithelial cells. Now a question comes to mind: what promotes the secretion of the chemical messengers? Some cytokines are constantly secreted at a low level. Most are secreted only when cells become activated as part of the response to infection. Cytokines are secreted at extremely variable levels. The two immune responses that we mentioned before, innate and adaptive immune responses, show differences in their cytokine secretion. Since we are talking about a network, of course, the cells need to communicate with each other in order to convey signals. There are three modes of cytokine-mediated cell-to-cell communication. Cells of the immune system communicate through the exchange of secreted cytokines. Depending on the spatial location and identity of the cytokine-consuming cell, such communication can be autocrine, paracrine, or endocrine. In long-distance endocrine signaling, signals are produced by specialized cells and released into the bloodstream, which carries them to target cells in distant parts of the body. Signals that are produced in one part of the body and travel through the circulation to reach faraway targets are known as hormones. Often, cells that are near one another communicate through the release of chemical messengers, that is, ligands that can diffuse through the space between the cells. This type of signaling, in which cells communicate over relatively short distances, is known as paracrine signaling. Autocrine signaling means the production and secretion of an extracellular mediator by a cell followed by the binding of that mediator to receptors on the same cell to initiate signal transduction. Now, in the case of the immune system, it mainly uses the paracrine and autocrine mode of communication. Now that we have discussed the modes of communication, let's jump into how are the cytokines stimulated. When there is an inducing or stimulating signal, specific cytokine genes are activated. This would ultimately lead to cytokine-mediated signaling in the recipient cell or the target cell, and the target cell would have a variety of biological effect due to this cytokine. It would either divide, it can differentiate, or even sometimes it can undergo apoptosis. So a variety of outcome could be achieved from cytokine signaling. The cytokine signal is received by receptor. Cytokine receptors are also often only expressed transiently. The complete IL-2 receptor is only transiently expressed by activated T cells. Again, this mechanism has evolved to prevent inappropriate activation of the immune system. Two more important features of cytokines that you should familiarize yourself with are redundancy and pleiotropism. Redundancy refers to the fact that, in general, several cytokines secreted during an immune response have very similar properties. For example, TNF and IL-1 have similar effects. These cytokines synergize with one another. In other words, the impact of both cytokines being secreted together is greater than the sum of the effects of the individual cytokines. This is important clinically because attempts to block the effects of cytokines may not always guarantee clinical outcomes. Anti-TNF monoclonal antibodies are successful at preventing joint damage in rheumatoid arthritis, for example, but do not completely prevent disease because IL-1 is also mediating damage. Pleiotropism refers to the fact that many cytokines affect several different types of cells. This is also clinically important. The antiviral effects of interferon alpha are used to treat hepatitis B virus infection. But interferon makes patients feel unwell because it induces an acute phase response. That is all about the general characteristics of cytokines. In the next section, we will talk about the classification of cytokine family and their functions.